Hi everyone, my name is Catherine and I am a third-year biology major conducting research at the Peng Lab in the Jules Stein Eye Institute. Today I will be presenting my research entitled Understanding Retinal Circuit Formation and Development through Cell Type-Specific Profiling of Cell Surface Proteins in Mice. Our vision works by light entering and traveling to the back of the eye where it converges at the retina. Information is passed through neurons at each of the different cell layers and ultimately sent to the brain through the optic nerve. This process is what allows us to see and perceive all the little things in the world around us. To briefly go over the structure, the retina consists of 3 nuclear layers, including the ganglion cell layer, internuclear layer, and the outer nuclear layer, as well as 2 synaptic layers, including the inner plexiform layer, or the IPL, and the outer plexiform layer, also known as the OPL. Within these layers, the mammalian retina harbors hundreds of cell types that assemble and synapse together, forming one of the most complex neural circuit systems. This is depicted here in this image to the left. On the right-hand side, we could see a more simple diagram of how the photoreceptors, which are labeled in purple, and horizontal cells in red, are connected, specifically in the OPL, which is a large part of what our research focuses on. Photoreceptors consist of rods and cones that allow us to capture and process light and color. Meanwhile, the horizontal cells modulate the output and integrate signaling from these photoreceptors. From this knowledge, we developed the research question, how are cell type-specific connections achieved in the mammalian retina? Our goal is to determine differences in cell surface protein expression among retinal cell types at various developmental stages, and through this, we hope to better understand the dynamics of cell circuitry in the retina. Studying how these retinal circuits form and develop in early stages is important because it provides insight into how our vision works and the many different factors that play a role in regulating it. For our methods, retina samples are obtained from several transgenic mouse lines, each expressing the enzyme Cre recombinase under a cell type-specific promoter. Each mouse was then crossed with transgenic mice expressing HRP at the plasma membrane to label all cell surface proteins through a biotinylation reaction. This method is known as IPL, which stands for in situ cell surface proteome extraction by extracellular labeling. As outlined here in this image, the idea is to use a transmembrane protein with the HRP enzyme in the extracellular portion. HRP then catalyzes tagging of cell surface proteins with biotin XX phenol or BXXP. After this reaction is complete, we can see that the cell surface proteins have been successfully labeled with biotin, as shown in green. Following optimization of experimental conditions, collected tissues were labeled through this process of immunofluorescence staining and subsequently imaged using confocal microscopy. Samples that showed successful biotinylation of surface proteins were then evaluated through proteomics analysis and gene ontology. Shown here are images of selected stainings done on 3 of our mouse lines. For the IPL, we have the VGLUT2 HRP line targeting labeling of retinal ganglion cells in P5 mice, and the CHAT HRP P6 mice localizing to starburst amacrine cells. Meanwhile, in the OPL, we use the HRGP line to label cone photoreceptors in P9 mice. For all of our labeling experiments, we look to see that the biotinylation was specific and efficient, ensuring that the HA tag for HRP and the neutravin stainings for each Cre line were consistent and localized. Following successful biotinylation, we conducted proteomics analysis on the tissues we collected in order to gather data on the specific proteins that were being labeled and enriched through our experiments. We mainly focused on the OPL lines consisting of CX57 for horizontal cells, Rho for rod photoreceptors, and HRGP for cone photoreceptors. Across all 3 cell types, comparisons between control and experimental samples collectively revealed upwards of 2,000 differentially expressed proteins. Additionally, gene ontology results yielded many proteins belonging to categories such as cell adhesion and signal transduction. After proteomics analysis, we derived a list of candidate cell surface proteins for each cell type, allowing us to delve deeper into studying the role of individual proteins found to be enriched and their respective functions in mediating synaptogenesis. Ongoing analyses have focused on validating top protein candidates through additional labeling experiments. One such protein is protocadherin 16, or PCDH16, which is a calcium-dependent cell-to-cell adhesion molecule. Here we used P11 mice from the CX57 line to target PCDH16, which has been labeled in pink, and we can see that it largely localizes to the horizontal cells as predicted by our data. However, one limitation is the fact that there is nonspecific green fluorescent protein labeling, and therefore our experiment could perhaps benefit from labeling a second time on a different set of tissues from the same line in order to improve labeling results. In terms of future directions, we will continue validation experiments for PCDH16, perhaps incorporating other techniques that may allow for better labeling of proteins, such as treating the tissues with antigen retrieval prior to staining. We have already run some experiments using heat-induced antigen retrieval, but found it to be unreliable at times in preserving tissue integrity, and so in the future we might also consider trying an STS antigen retrieval protocol instead. Additionally, another future direction would be to induce loss of function and/or gain of function for the PCDH16 protein in mice and see how that might affect the synaptic connections formed in the OPL. Finally, we will continue with validation experiments for other protein candidates selected for the OPL lines, and also work on identifying the best candidates to focus on for the IPL through ongoing literature search. I would like to thank Dr. Jorge Avila and the UCLA Undergraduate Research Fellows Program for supporting me and helping me to become a better researcher. Thank you also to the Anna and Kenneth Taylor Endowment and the James and Meredith Henry Endowment for their generous support. Here are my references, and that is the end of my presentation. Thank you so much for watching.